project was algae and biofuels and how you create biofuel from algae. Now, it's no secret that in today's world and in the modern world, as we look around, we see that there's a lot of problems in the modern world with fueling the world. Namely, the fact that current methods of fueling the world, moving cars and all sorts of things from place to place, uses petroleum and fossil fuels. But that's neither sustainable nor is it eco-friendly, because the simple facts of the matter are eventually we'll run out, one, and two, it damages the ecosystem. So we set about to see if we could try to create a biofuel using algae. And so we wanted to see how we could increase the lipid content in order to do that, in order to set about increasing the ability to create biofuel, because the essential part of biofuel is the lipid content. And so we tried to do this by creating by adding carbon dioxide and creating a deficiency of nitrogen. And that basically just stresses the algae to increase lipid production, because they're going to be more likely to store resources in the form of lipids if they're stressed than when they're unstressed. So this is a little bit about our procedure. Our first thing that we had to do is we had to actually set up and grow the cultures, which requires, each culture required its own specific media. For both the Chlamydomonas and the chlorella required separate medias and separate procedures. So we had to set up these cultures first and then expand them as time went on. So this is basically just the lab procedure and how we went through that. So we had to add the media, we had to add the culture, and there's obviously much more media than culture because the culture grows into the media. And then you have a loose cheesecloth cork to keep the system relatively sealed, and we labeled them, of course. So basically we looked at a variety of different factors. We looked at the effects of carbon dioxide on them, like adding carbon dioxide into the system. And when we did that, we also tried to control for pH change, because we realized when we were setting it up that the addition of carbon dioxide would change the pH. So we had to block for that variable. And in so doing, we added a pH buffer, to reduce the change in pH in some of them, so that we could compare and see, is it the CO2 that's doing it, is it the pH change, is it both? So we had to expand the experiment and set up multiple different cultures that way as time went on. And this is just our spectrophotometry, which is basically what we used to just see how the algae was growing as we were going through. And so the algae seemed to all be growing pretty well as time went on, with very few bumps in the road. So this is a look at our greenhouse setting. That's our algae on the rocker, where we put them before and after we add the CO2. That right there is a CO2 tank, and that was our method for adding CO2 into the system. It's closed at the top. The CO2 flows from the top into the system, into the algae, and then back out, and then we stopper them. And this is flow cytometry. This is how we checked our results. This is how we monitored and managed and checked the lipid content inside of cells. So what we did is we used flow cytometry, and especially the flow cytometer at Dendrion, to check this. We basically stained the dye. We dyed the algae. We stained them with a Bodivi dye, which basically stains the lipids. And so as they went through the flow cytometer, they would fluoresce when they had lipids. And we created a baseline for that. So we had the baseline without the stain, and then with the stain, so that we could see how much actual fluorescence there is as a result of the lipid content increasing, as opposed to just the natural fluorescence of the cells. And so the flow cytometry was a very powerful tool in finding out the lipid content to check to see if our experiment was working. And this is just a bit of a background onto how we got onto algae. Last year we did some projects regarding algae and cancer and algae and food. And so this year, and that would be here increasing protein for food and such, and over here we looked at radiation and its effects on the DNA of algae. And so this year we tried to take the algae in a new direction. We tried to take it in the direction of biofuel and see what we could do to increase the lipid content so that we could refine biofuels, as opposed to increasing the protein content to create food. And this is some background on how flow cytometry works. Flow cytometry basically works because you get laminar flow as things are flowing through the pipettes and into the interrogation point where lasers go, and it basically forces the cells into a single file line, and the laser bounces off of the cells and refracts into the data collection points around the edges. And some of them are dedicated fluorescence detectors, some of them are forward and side scatter detectors, which detect basically size and density. And so the density is basically cell health, like how many organelles are there, how dense is the inside of the cell, and the size is basically how much did the cell grow, how big is it. And so these are our results we got from the flow data at the end of the session, at the end of our thing. So this is Chlamydomonas, and that's our control. This is it with CO2, this is it with CO2, and that's it with CO2 in the buffer. Now, if you look at the Chlamydomonas, with these two in particular, just the CO2 without the tris buffer, we did get a very high lipid concentration on the y-axis. But if you look, the quality control on Chlamydomonas has to be much, much higher, which is something we'll point out in the conclusion later as an affecting point. Because as you can see here, the size of the algae is represented on the x-axis is very, very small, with the concentration being very close to zero, which means we had a lot of dead algae, a lot of small algae, a lot of unhealthy algae. And so the quality control has to be much tighter so that you get a graph that looks like this with your algae production, rather than a graph that looks like this. And then with tris, it was kind of an intermediate range. You kind of had the full spectrum all the way, not as good on lipid production and not as bad as the second graph on size. But this graph right here is the best overall out of all we've seen with regards to cell size and lipid production. And this is our chlorella. And chlorella has a lot of the same problems that we saw before. But if you look at the chlorella graphs, their values are much lower, being concentrated here. This one has a little bit of a high point there, but the majority of the data is concentrated in this section right here, which is lower than Chlamydomonas' high point. And if you look at this one, which is more like our baseline for chlorella, the chlorella with CO2 is significantly lower than the Chlamydomonas. Even though it's much healthier, it's much easier to grow, it requires much less quality control, what we saw is the results were that you would end up with a lower lipid concentration. So it's easier to grow, it's faster to grow, it's a lot easier to maintain going forward, but it doesn't give you as good of results as high quality fuel and as many lipids as the Chlamydomonas would. And so our conclusion was basically we wanted to see which species would produce more, and we wanted to see the higher levels, and we wanted to check like the effective pH. And basically we found that the pH is an important factor in raising algae levels. The ones that were buffered did not produce as high lipid concentrations as the ones that did not have buffers. And so we know moving forward that you don't want to buffer your algae fuel type samples when you're growing them, because that pH change is actually a significant factor in increasing the stress of the algae. In addition, when we look at it, we see that Chlamydomonas is much better for a high quality fuel, but it's also much harder to control, it's also much harder to grow. So you have a thinner margin of error and you have to have tighter quality control on Chlamydomonas, which would make it in an industrial setting more expensive to grow than the chlorella. And the chlorella still got excellent results. Its results simply weren't as high though as the Chlamydomonas, but it was much hardier and much easier to grow. So you kind of see these two directions in biofuel between the two of them. The chlorella is really suited to like low quality, mass-produced consumer fuel, and the Chlamydomonas is more suited to things that would require a higher octane level, a higher level of potential energy inside of it because it has more lipids concentration, and because with those higher quality fuels and higher quality needs, you have a bit more budget to play with in terms of making the cost more expensive. And so this is fatty acid synthase, and this is basically the protein that's responsible inside. This is a mammalian fatty acid synthase. So it's a model, but it's not an exact model of what's in algae, but they are very similar just as like a comparison point. And this is basically what's responsible for creating fatty acids, which are the lipids. And so it's important to do the experiment because our goal is to increase those fatty acids. And in order to do that, you have to kind of have an understanding of what does that in the cell, and that what does it in the cell is fatty acid synthase. So references.